So this is a general review of what we did at the end of the first semester, Body Systems and Homeostasis. There are a few things that we ran out of time to cover that I'll be going over here. So those will be helpful for you to have. So first, let's talk homeostasis. So these are copied and pasted directly from the thing that tells me what to teach you guys. And so one of the things I'm supposed to have you guys understand is that organisms have areas or compartments that perform specific functions related to energy, matter, and other functions to keep organisms alive. And together, these parts work together to keep our bodies functioning. Those compartments that organisms have, it depends on if you're unicellular or if you're multicellular. So some of the functions that these compartments do is they exchange gases. So we have a couple of examples here. For example, I have gas exchange in one-celled organisms. That is very simple. They just do that through their cell membrane or their cell wall. Amphibians do so through their skin. Notice that it has a layer called epidermis, and there's CO2 and O2 coming in and out of that. That is actually the skin of a frog. Then echinoderms are things like sea stars, and they exchange gas through their outer surface as well. So do insects. Fish are some of the organisms that have something that is an organ devoted to exchanging gas. They have gills, so blood vessels run through these gills, and as water runs across the gills, oxygen is absorbed, CO2 is released. And then mammals have more, I don't think it's more developed. It's different because we have to allow air in and out of our lungs, whereas fish just have water running through their gills. So we have lungs that are going to exchange oxygen and carbon dioxide for us. There are a series of tubes that lead to finer and finer tubes until they end in these gas-filled sacs called alveoli that are very heavily surrounded by blood vessels, so that the gas exchange can occur. You'll notice that as soon as you get above the level of a single cell, not only do you need a way to exchange oxygen and carbon dioxide, you need a way to transport that around the body. So respiratory systems and circulatory systems are very heavily linked. Then I also included a diagram of a leaf on here as well, because plants are organisms too. We need to remember that when we're looking at body systems, because you may be required on the test to make comparisons between plant and animal body systems and how they function. So plants exchange gases through holes in the bottom of the leaf called stomata. A single one is a stoma, and they're just gateways into or out of the leaf, but the plant does have the ability to close and open these stomata as well. So it's a little like a mouth on the underside of a leaf, so they have control over it. That's also where water comes out during transpiration. Circulation of fluids happens in different ways as well. I have a number of different circulatory systems up here. What you'll find that they all have in common is there are systems of tubes, and those do the actual work of carrying around the blood. And then there is usually some sort of pump. You need to use energy to move this stuff around the body. It must constantly circulate. If it's not moving all the time, it's not able to adequately exchange nutrients and gases. So there's usually a heart or something heart-like that's going to pump. You do have some organisms that have more than one heart. So the earthworm that you see up there actually has like 5 or 6 little mini hearts that each pump blood. Something like an insect has a more primitive system where the blood can leave the circulatory system, and it's not actually technically blood, it's another type of fluid. We're not going to go over the name, but it collects and it drains from the body into the heart, and then from the heart it gets pumped out, and then it leaves tubes of the circulatory system and just goes out and bathes the surrounding tissues. You can see that as you move from insects, which have a more primitive circulatory system, to something like a worm, you get that evolutionary development where you now have a closed circulatory system. So your blood is going to stay in there instead of your blood just circulating around the cells. That becomes even more complex when you look at the circulatory system of fish, amphibians, mammals, and birds. Not only is it closed, but you start seeing these other loops. So on amphibians, mammals, and birds, we have what's called a pulmonary circulation, which is what goes to our lungs, and then you have the systemic circulation, which goes to the rest of your body. So one of those is designed to pick up oxygen and drop off carbon dioxide, and the other one is taking it around to the various tissues in the body. Plants also have a vascular system, but they do not have anything like a heart, so they don't have any pump. Instead, they rely on changes in water potential to drive water up. They have to have water evaporate out of their leaves by transpiration. If they don't, the water that's in their vascular system will just sit there. And that means that once any nutrients or sugars are used up in it, there won't be any more until you get that water to move. So that is actually an evolutionary adaptation of plants to make sure that they can transpire in any environment without losing all of their water in their body. Because if they can't transpire properly, they cannot carry nutrients around. Any questions, comments, concerns? I know there's a lot of stuff on some of these slides, but as it is, this is like 105 slides, and I was trying to make it less. There are some similarities in digestive systems of various organisms. Plants don't have a digestive system because they make their own food, so they don't have to worry about getting external food and digesting it. They'll just break down some of their starch storage and start using glucose. Animals, on the other hand, are all heterotrophs. We all take in food from the outside world, and then we have to break it down and then get those nutrients out to the body. So there are two major types of digestive systems. One type is a single opening where you have a mouth-anus combo and you have some sort of stomach cavity. So everything comes in and goes out through the same hole. Kind of nasty, but those are mostly those are just invertebrates. And even in some invertebrates, you start getting the more complicated two-ended digestive system where you have a mouth at one end and an anus at the other end, so that you can, the problem is that in something like a cnidarian, which is what you see up there with the mouth-anus combo, they'll take in food, but they cannot take in new food until they're finished digesting the old food, because they have to empty out their stomach before there's room for more food. So if they're in an area where there's plenty of food, they might have to eat, wait a while to digest, poop it out, eat, wait a while to digest, poop it out. Whereas if you have a two-ended digestive system, you can, nastily enough, eat and poop at the same time, like this little bunny does. Or a sea cucumber. I did not know they had two-ended digestive systems. Ah, okay. Well, just like that worm that's up there, it's a straw. And then that's how the digestive system evolved first in animals. It was just a tube going through. Usually there was like these slight larger areas like bulges, like for the primitive stomach. And in our digestive system, that's now evolved so that we have a distinct stomach, which you can see in things like the shark, salamander, lizard, etc. up there. You then have that tube connect directly to the small intestine, which then connects directly to the large intestine. And then as things got more complicated, you ended up having other accessory organs that are not part of that main tube, things like the liver and the gallbladder, whose job is to make digestive enzymes and drop it into that digestive canal. Unicellular organisms don't have to mess with this. They just have lysosomes, so they take in food in a vesicle, lysosome it up, and then release the waste. So temporary structures only. Excretion of waste is fairly complicated. If you look at the structure of a kidney, you will probably look at it and go, holy crap, I don't want to look at that anymore, because it gets kind of complex. But at its heart, what it is its blood comes in, you filter out any of the waste products like urea, sometimes, well, only if you have malfunctioning kidneys, sometimes proteins can come out through there, but any metabolic waste products. So this is taken out from your blood. If you eat something and then you excrete it later, that is not the same sort of waste product that comes out in urine. So urine is mostly like molecular stuff. That's why it's all dissolved in a liquid. There's no solid in there. And so what will happen is they'll run blood through tubes that will be alongside a tube that will collect urine. And the urine is any excess water as well as these waste products that are dissolved into it. And usually your kidneys will work in such a way that this stuff will will diffuse down its concentration gradient and then naturally move to the urine tube. So you'll have a urine collection tube, and then that'll lead to an exit. So you have something like the annelid that's up there, that's a roundworm. It doesn't have a single excretory system. It has a bunch of those, what are called nephrostomes. You don't need to know the names. You just need to know that nephro means kidney. So anything that says nephro or reno has to do with the kidney. Or ure, like urine or uric acid. And so they have a bunch of those. They don't have a single kidney. They have like in each segment, they have a separate little mini kidney. And as you go to more and more complex organisms, those mini kidneys get more and more complex until you get to something like us, where we have a very complicated kidney system. If you take anatomy and physiology, you'll learn about that in more detail. All right, so these organ systems have to cooperate. No organ system is independent of the others. Each of the organs in an organ system have to work together. So for example, both the stomach and the small intestine participate in digestion, but they do different parts of it. The stomach is a lot of mechanical digestion. It physically breaks things into smaller pieces. And it does have some digestion. It starts digesting like proteins, and you actually start digesting starch in your mouth. The small intestine, though, is going to finish that digestion, and it is going to absorb nutrients. The digestion is going to happen mostly with enzymes, and these enzymes have different pHs at which they function better. So the stomach and the small intestine have dramatically different pHs. If you take an enzyme from the stomach and put it into the small intestine, it will not work because the pH is wrong. And vice versa. So each of them has slightly different conditions to allow for digestion of different molecules. The kidney and the bladder work together to help in excretion. So the kidney is what actually filters the blood. It collects urine. That urine drips down a tube called the ureter, and that is held in the urinary bladder until you are able to excrete or pee. And that is a consciously controlled muscle. That's why you are not constantly dripping urine. You're able to hold it until you get to a place where you can release it. Think about how inconvenient it would be if you had no bladder. And people who have like bladder problems or have kidney problems, they might have to have a catheter, which is they stick a tube up the urethra and collect the pee that's constantly dripping down. Not pleasant. In plants, you don't generally think of plants as having organs, but you can think of like the roots, the leaves, the stems, each as individual organs, and they all have to work together in order to keep the plant alive. So the roots get in all the nutrients from the soil. They might collect they'll collect water as well. That's got to go through the stems. The stems are important because they keep the plant not only upright, but they help the leaves get closer to the sun. And the sun and the leaves use all of this stuff in order to produce the sugar that feeds everything. All right, so systems are also going to have to interact in order to keep us alive. So I've already stated that the respiratory and circulatory systems are very closely connected. Without the circulatory system, the respiratory system would not do any good for us. So you'll always have those two systems linked in whatever animal you're looking at, or you'll have very heavily vascularized respiratory tissue. There's going to be a lot of blood vessels in there. The nervous and the muscular system work together. So there is something called a neuromuscular junction wherever a neuron hits a muscle cell. And when a neuron fires, it's going to signal the muscle cell to contract. And that is going to be what happens when you are moving a muscle. So without that signal from the nervous system, though, the muscle does not contract. Plant vascular systems are necessary to bring materials to the leaf. They're what bring any mineral nutrients, water, and water is needed for photosynthesis as well as transpiration. Then those leaves are going to be the site of making glucose, and to support all the other cells in the plant, they are going to have to transport that through the vascular system to everything else. Okay. All right, so let's talk feedback. There are two major feedback mechanisms that help us maintain homeostasis, or in some specific cases, disrupt homeostasis for a particular purpose. So negative feedback systems are designed to keep homeostatic conditions. They will eliminate any variables or changes and return you back to the original condition. So a negative feedback loop usually has some sort of set target. A negative feedback loop will keep you at that target point, and anytime you have any imbalances, it will help correct it. So what we see here is basically a diagram. You have an imbalance. There's some change that's detected in your body that is going to be sent to various, like the brain or the endocrine system, and that's going to cause your body to do something, some sort of action via an effector, which could be like muscles. So if you're cold, your muscles are directed to start shivering, or it could be glands, like your sweat glands are directed to start making sweat when you get too hot, and that restores you back to your original condition. So negative feedback loops, the negative part is they're negating any change that's happened. An example of that is temperature regulation. You get too hot, you're going to sweat. You get too cold, you're going to shiver. There are a couple of other ways that we regulate body temperature. We do this very actively because we are endotherms, meaning we're warm-blooded. So we spend a lot of energy to do that. There are various reactions that plants have in response to like low water conditions. There will be some that are long-term, some that are short-term. A lot of it has to do with osmosis. So they can change osmotic conditions, so that way you end up encouraging water to enter by controlling your solute concentrations, or you can also open and close stomata for a short-term fix. There are positive feedback mechanisms as well. Positive feedback mechanisms are going to not only maintain a change, they're going to make the change more severe. So you have your normal homeostatic conditions, and you maintain those with negative feedback. If you want to disrupt homeostasis, you do that through positive feedback. It's an amplification signal, so it's going to make whatever change you have stronger. So this is the general way that positive feedback loops work. You have some sort of stimulus, and that stimulus is a change in body condition. That triggers a response that's going to increase that change in body condition, which is going to further stimulate the body, which is going to respond by increasing the change. And you may think, well, when do you want to change something that bad? If you're trying to have a baby. So this is the biggest example of a positive feedback loop. What ends up happening is the head of the baby moves into position, it presses against the cervix, and that sends signals to the brain, which then releases a hormone called oxytocin, which is carried in the bloodstream that triggers the uterine muscles to contract. That pushes the baby's head harder against the cervix, which sends more signals to the brain, which stimulates the release of more oxytocin, which triggers the uterine muscles to contract even harder until eventually the baby comes out. Because you don't want to maintain homeostasis of keeping a baby inside. Is it good for the baby to stay in the womb too long? No. If the baby is not born at the proper time, it could die. Yeah. So that's why, you're not supposed to hold it in if you're feeling like you're going into labor. No, you can't. And there are other examples. So, for example, lactation also has to do with oxytocin. So what happens is an infant suckles at the nipple. That triggers the mammary glands to produce more milk. by a series of hormonal changes involving oxytocin as well as the brain. That will release milk, which will trigger the infant to suckle more, which will produce more milk. Eventually, if you are breastfeeding, if you stop breastfeeding without being triggered by the baby suckling, you'll actually stop producing milk. And that's how you wean an animal off of breastfeeding. My cat happened to have accomplished that by biting her children on the head whenever they would try to nurse. I told my mom, thank you for not doing that to me, and if you did that to me, I'm glad I don't remember. There are also these positive feedback loops in plants as well. So when fruit ripens, that's actually the result of a positive feedback loop. So what happens is one particular fruit on a tree ripens. Well, that fruit produces something called ethylene. Ethylene is involved in signal transduction or cell signaling, and what it does is it turns on a whole host of genes inside of a fruit, inside the cells of a fruit, that will cause it to ripen. So that'll break down starch, release sugars, usually a color change will be involved. It may release smells to attract whatever animals eat that fruit, and that is going to make those apples then produce more ethylene, so all of the fruits in the surrounding tree will get riper and riper. In fact, what they do sometimes in supermarkets is they will, or when they're transporting fruit to supermarkets, they can't pick it when it's ripe, because if they do, by the time it gets to the grocery store or your house, it'll be overripe, it'll have gone bad. So they usually pick these while they're still green or unripe. And then once they're getting closer to the supermarket, they'll pump in some ethylene and ripen everything up. So that's how you get fresh fruit. Blood clotting is another example of a positive feedback loop. This is also something where you want to increase a change. The change is sealing that hole in the blood vessel. So when a blood vessel has a tear in it, that is going to attract platelets. Platelets are going to bind to that area and release chemicals that attract even more platelets. And they'll keep doing that and attract more and more and more and more platelets until finally the crack is sealed. No more blood is coming out, and that stops the positive feedback loop. Okay. When you have something that disrupts these feedback loops, though, that is going to end up usually causing diseases. So one that everyone should be pretty familiar with is diabetes. Diabetes is usually you are not either producing or responding to insulin properly. So what ends up happening is usually when your blood sugar is high, your body releases insulin. It actually attaches to an insulin receptor, which, by the way, is a receptor tyrosine kinase. No. Okay, that sets off a set of signals that is going to then cause glucose transporters to be sent out to the membrane, so they'll start picking up glucose from the blood. Well, the problems with type 1 diabetes is there is no signal, no insulin, so the insulin receptors are not going to be turned on, they're not going to send signals, and that glucose is not going, those glucose transporters are not going to be sent to the surface of the cell. Type 2 diabetes, you do have insulin, but something is wrong with the insulin receptor or with the cell signaling inside of the cell. So the insulin is out there, it is sending messages, but you are not receiving those messages properly, so the glucose transporters don't go to the cell membrane and you're not taking in any glucose. You can also, if you have, for example, if you have an antidiuretic hormone, there are things called diuretics. They make you pee more. Antidiuretic hormone is supposed to make sure that you are getting enough water if you're dehydrated. So it's going to have a couple of effects, one of which is to make you thirsty, so you'll go drink. You have some osmoreceptors, which detect your osmolarity. You also have signals sent to the kidney, stop taking water out of our blood, we need to conserve it. And if there's a problem with that, you could end up having somebody who dehydrates, or they end up urinating too much, so that would be a problem. There's something called Graves' disease, where you have your thyroid hormones producing way too much hormone, or they're overstimulated, and what will end up happening is, due to those overstimulated thyroid hormone receptors, you will actually have something called hyperthyroidism. Your metabolism is very sped up. You might have a goiter, which is this big bump in your neck because your thyroid gets bigger, and there are a number of other symptoms. Okay, so some of these homeostatic control systems are conserved between plants, between animals, and even bacteria, so that supports the idea that we all evolved from the same ancestor. So what you can actually see is this is the excretory system, the evolution of the excretory system. So you'll see that it started out fairly simple. There is similarity between even the excretory system of insects and so vertebrates and invertebrates, insects and fish. And then you can also see how these kind of got modified as you evolved into more complicated organisms. So that's some evidence of common ancestry as well. There's osmoregulation is a very important thing for different species. Any species, you need to make sure you have enough water. If you live in freshwater, for example, and you're a freshwater fish, that means that water is going to constantly be coming in. So what they do is they will not drink water, and they will pee quite a bit because water comes into their body from the pond or lake or river. A marine fish, on the other hand, is surrounded by very salty water. If they don't regulate the osmolarity, then what will end up happening is water will exit their body. So what they do is they are constantly drinking water and exporting sodium. And bacteria have to do this as well. Certain single-celled protists called paramecia actually live in freshwater, and they don't do what the freshwater fish do. They don't pee. Well, they kind of do. They have a contractile vacuole that absorbs all the water in their body, and then they squeeze it to remove any excess water at the end. This is also, you can look at evidence of common ancestry in the osmoregulation in aquatic and terrestrial plants. Plants evolved on land. Things that never left the water are things like algae. So when plants, land plants, terrestrial plants, ended up moving back into the water, they were actually having to evolve adaptations to go into the water. So you have to have water constantly moving through a plant. That happens through stomata in terrestrial plants. Aquatic plants have changed, so they can actually release or absorb water through the entire body surface. All right, thermoregulation is also something that provides evidence of common ancestry, so you can actually track this as you look through some things that are more ancient, evolutionarily speaking, versus us. So what you see at the bottom on the right-hand side where it says organisms, on the right-hand side you see a phylogenetic tree. So it's showing how these different mechanisms of keeping warm or maintaining temperature, where they evolved, when they evolved, and so you can kind of see some of the differences that in the mechanisms by which we keep ourselves warm. So for example, let's see, if you live in the water, water temperature tends to remain fairly constant, but temperature on the land fluctuates quite a bit. If you move from water to land like the first vertebrates did, you have to find a way to regulate your body temperature because there's no water there to do it for you. So you'll see some modifications of the natural homeostatic mechanisms that were in fish, which, you know, have their temperature regulated by the surrounding water, and us, who have to regulate our own body temperature. So you'll see, like, for example, on the left, you have a diagram. It's a graph of the body. Ooh. Can somebody get me a napkin? That's what you were doing. You see the relationship between the body temperature of, yeah.
That's a lot.
There's one on the floor too. Here.
This is like Reese's Cups.
Yeah.
Like the cereal.
Yeah, I know.
Oh no.
And so you see a river otter no matter what the outside temperature is, it is still able to maintain a constant body temperature, internal temperature. However, a largemouth bass, which is ectothermic, is not. Its body temperature, fluctuates with the outside temperature. That's what happens when you feed them. I think that's it. You also see some difference in temperature regulation in different types of fish. Some fish are going to actively, they're called hot fish, and they are going to regulate their temperature a little better. What they use is something called counter current exchange, where blood that's flowing out to the outer side of the body where it's colder is going to run past the blood that's coming from that cold water area, and so it's going to kind of even out the temperature so that they don't have as many extremes in temperature. All right, organisms have various mechanisms to respond to changes in their environment. So in plants, you have two responses to sunlight called photoperiodism and phototropism. So phototropism is growth towards, or a tropism is growth towards or away from something. With the photo part, what does photo mean? Photo means light. Phyte means plant. So phototropism is when you grow towards or away from light. Being a plant, what direction do you want to grow? Towards the light. So this is all mediated by a particular hormone called auxin. Auxin is released on the dark side of a plant. And what it does is it's a signaling protein that is going to tell a plant's cells to grow longer. So since it's on the dark side of the plant, it's going to make those cells longer, and that's going to cause the plant to bend, and it's going to bend towards the light. So that way it will have the best chance at performing photosynthesis. On the other hand, there's a reaction called photoperiodism. That has to do with how much sunlight you're getting and how much nighttime darkness you're getting. That's going to help control when flowering happens. Why would it be important to control when you flower if you're a plant? When's a bad time to make flowers? In the winter. Most plants, if they flower in the winter, they are going to be destroyed or damaged by frost or ice or anything like that, and they are not going to be able to reproduce. So plants have these mechanisms that have evolved. So they only flower when the nighttime reaches a certain length, or when daylight reaches a certain length. So you have some that are short day, long night plants. If you have a short day, if you have daylight only for a short period of time, they'll flower. If you have any light interrupting the darkness, they will not flower. You have long day plants that will only flower if you have a longer day and a shorter night. And this is of some importance if you have environments like a city where there is never nighttime, never full darkness. That can affect how these plants flower or don't flower, which could be a bad thing, especially if you're trying to grow like food plants. Okay. Then there are directional movements in animals. You have taxis and kinesis. Kinesis is just random movement. So what you see in that diagram on the right is kinesis of a roly-poly. It's wandering around until it finds something that is useful, in this case, a moist area so that it can not lose water. On the other hand, taxis is usually towards or away from something. It can be positive taxis, where you're moving towards a stimulus, or negative taxis, where you're moving away from it. And you can also have different prefixes on the word taxis. So for example, chemotaxis is attraction or repellent from a certain chemical. So unfortunately, mosquitoes have a positive chemotaxis to CO2. So if you're outside and you're breathing, they're attracted to you. And that's how they find you to bite you. Or if you're like me, that's how they find you and bite the crap out of you. Bacteria do the same thing. They have receptors on their surface, and depending on which side of the cell the receptors are activated on, let's say this is a chemoattractant, those receptors, which you see in the surface of the membrane, if they're activated, the bacteria will swim in that direction. With a chemorepellent, whatever receptors are activated by that molecule will trigger the bacterium to move in the opposite direction. You also have patterns of activity. Some animals are asleep during the daytime, and those are nocturnal animals. They're awake at night. They have adaptations to allow them to hunt better than if they're a predator. So, for example, they usually have very good ears since their eyes might not be as useful because it's dark. On the other hand, diurnal animals that are awake during the daytime, like ourselves, tend to have pretty sharp vision because we're relying a lot on visual cues. You have a circadian rhythm. That's your natural sleep-wake cycle. There is a part of it that's genetically mediated. If I stuck you in complete darkness 24 hours a day, you would still sleep for a certain amount of time, be awake for a certain amount of time. Same if I kept you in constant light. And they've actually done experiments like that, where somebody's in a room voluntarily, they're kept there with constant light, and scientists were like, okay, do you sleep in a regular pattern? And they did. We even saw an FRQ about that too. On the other hand, we also can reset our circadian rhythm. Like if you ever travel to another part of the country that's far away, or to another country and you're on a different time zone from them, like if you go to Europe or if you go to Asia, I don't know if you guys have been, I have, and you know how you get jet lagged? That's because your circadian rhythm is no longer the same as the light day cycle of the place you're at. Your body is trying to respond to these outside cues and reset your circadian rhythm. It takes some time for it to do that though, so in the meantime, you're sleepy at all the wrong times. Yeah, but if you stay in that location long enough, you'll adjust. Usually takes, if you're moving forward in time, I think it's a little easier than backward in time. It's all unpleasant to me. All right, let's talk about the immune system. So we have different types of immune systems in different organisms. Plants, invertebrates, and vertebrates all share something called the non-specific immune response. So this means it doesn't depend on what the pathogen is, they're just your general responses. So for example, we have lysozymes in our intestines. Invertebrates have that as well, so they just break down whatever goes into their intestines. So if you eat something that has bacteria in it, or if an invertebrate does that, then it will break it down. You also have certain peptides that are antimicrobial. They are designed to break down chemicals or shut down processes that happen only in bacteria. So they're not dangerous to eukaryotes like us, but they're dangerous to prokaryotes. And there's also just plain old phagocytosis. If you don't know if it belongs here, just eat it. No, that's apoptosis. Yeah, apoptosis is the cell suicide, and it actually has a role in the plant immune system. So plants don't have this roving like police force of immune cells like we do. Instead, what happens is each plant cell is its own defense. They have a lot of chemical responses that they can put into place whenever they have an invasion happen. And cells will, if they need to, commit apoptosis, cell suicide, to destroy themselves, so the pathogen will hopefully die with them. And that way it doesn't spread. So they'll be like, I sacrifice myself for you. In fact, you can see this sometimes with certain trees or other large plants. If they have something toxic that's in their system, they'll stick it in a leaf and they'll let that leaf fall off because they can regrow a leaf, no problem. We don't have that same luxury. If my pinky finger fell off, I would have a loss of function, which would be evolutionarily bad for me if I wanted to keep living and have some kids. Yeah, cancer's a little different because that's your own cells turning against you. This is, you've got an invader that you're trying to get rid of. Plus, for that, you're cutting something out, but it's not something your body is naturally doing. So, Vertebrate immune systems are more complicated. So we do have our non-specific defenses, things like the acid in your intestines. Your skin is the first non-specific defense. That's why if you have a cut in your skin, you're supposed to be careful with it, put some antibiotic ointment on it, because that's a breach in your defenses, and then an invader can get in that way. Oh, dude, again? Let me just bring me a wad of paper towels. Can you just bring me a wad of paper towels? Oh, good Lord. All over the place. Here, dude.
Oh no, he kicked it.
Oh, he almost fell. For anyone watching this video, we have a rabbit. He's pooping on my desk. And maybe me. Okay. You just bring me a wad for the next time this happens. All right. So we have a number of these innate, non-specific defenses. The physical barrier of our skin, chemical barriers are things like enzymes. There's stuff in our saliva, in mucus, in our stomach, there are acids. We have patrolling cells like macrophages that are going to phagocytose pathogens. They're going to destroy any infected cells. And then you have things like fever or inflammation. So I annoy my family very much because whenever I get sick, I only take cold medicine when I have to be at work. If I'm at home, I just let the fever reign. And the reason why is because I'm like, it's your body's own natural defenses. So I try to use them for my own good. Inflammation is also another way that your body deals with infection. So that's why, for example, all of my lovely cat scratches, if I don't treat them with antibiotic ointment quickly and cover them up, they'll get red and they'll swell up a little. So that my body can try and keep any bacteria that might have gotten in through that break in my skin from getting to the rest of my body. Okay. All right. So let's talk about the adaptive immune response. This is something that happens just in vertebrates. So we're special. It all has to do with antigens. And if you were in class today, you were probably like, frickin' antigens. So what ends up happening is your macrophages, which are your like hall monitors, that would be a really disturbing analogy, and I'll say why. So they're like your hall monitors, and they're wandering around looking for invaders. Except in the case of your body, if they find one, they eat it. So that would be like if the hall monitors ate you when they found you trespassing. Then they not only eat the trespasser, they break it into pieces and sell. And some of the molecules that are on the surface of that pathogen, whatever infectious disease agent you have, are going to end up being presented by that cell on their surface. So they are going to basically be a warning poster. It'd be like if that hall monitor, when it found you trespassing in a school, ate you, took a part of your body like your face, and started wearing it like its own skin and being like, look for this, destroy this. So like I said, a very creepy example, but it might help you remember. Oh, that's not even a tardy freeze. They don't wear your face. Okay. So these cells that are then going to display the antigen to your body are called antigen presenting cells. Their job, they're like wanted posters walking around. And they are also going to activate white blood cells so that white blood cells can then trigger their responses. So keep in mind, this is in response to a specific pathogen. Once that your body recognizes something as a disease-causing agent, it will be able to recognize it for a long time. In some cases, even for the rest of your life. It's specific to that particular pathogen. So let's say that I got infected by a cold virus 3 weeks ago, which I did. 2 weeks later, I got sick again. It's not because my immune system doesn't work. It's because I got infected by either a different virus or a different enough strain of the virus that my immune system couldn't recognize it. That's actually why we have no cure for the common cold right now. It mutates so quickly that our body may have a response to this strain that we got infected by today. If the common cold changes enough, in 2 weeks, we can get infected by another strain. So drink your orange juice, get some sleep. So let's talk about lymphocytes. There are 2 types, T cells and B cells. B cells are the antibody makers. So think about it, they're B cells. They make antibodies, which has a B in it. That's not what they're named for. They're actually named for where they're made. But I don't anticipate you having any questions about that on the AP test. Both T cells and B cells have receptors on their surface that can recognize particular antigens. So B cells and T cells are specific to a certain antigen. It could be a molecule that's on the surface of a bacterial cell. It could be a receptor on the surface of a fungal cell. It just depends on the antigen and what macrophage broke it down. So here's what happens, the sequence of events. Macrophage takes in something dangerous, breaks it apart, and wears parts of the invader like a gruesome trophy to help activate other cells. That is going to activate helper T cells. It's going to activate a specific helper T cell, the one that has a receptor that will bind to that. So there are other helper T cells that may not be activated by it. That's going to trigger those helper T cells to reproduce. So you're going to get a lot more that have that same receptor. And those helper T cells are going to then, in turn, activate B cells, which make antibodies, and cytotoxic T cells. B cells, they're just making antibodies. Cytotoxic T cells eat infected cells. So that's the sacrificing yourself for the good of everyone else. So helper T cells recognize a specific antigen. They're going to go and then activate B cells and T cells. Their job is recruitment. They are also the target of HIV, which is why your immune system, if you have HIV and it's progressed to AIDS, your immune system isn't functioning because you have an immune system. You've got your B cells and your cytotoxic T cells, but there's nobody giving them a signal to come to your defense. Oh, and some of these cells stick around as memory cells, and they can then respond much faster the next time you get infected by something. So we have 2 major types of immune response. Humoral is antibodies. You have antibodies in the bloodstream, that targets things. That targets the pathogens. Other cleanup cells, white blood cells like phagocytes, can come along and can eat those pathogens when you catch them. Cell-mediated immunity, though, is you destroy the cells that are infected. You sacrifice them to save the rest of the organism. That is done by cytotoxic T cells. They'll recognize the infected cells, and they will eat them. So, B cells are interesting creatures. Each B cell produces one and only one type of antibody. That antibody recognizes one and only one type of antigen. When a B cell is recruited by a helper T cell, that's recruiting the specific B cell that will react with that antigen. Notice up there you have 4 populations of B cells. Only one has the matching receptor, so that one is the one that gets activated. It starts reproducing like crazy. When it reproduces like crazy, some of those cells are going to go off and be saved as memory cells so that later on you can reactivate these cells much faster if you get infected. Other cells are those plasma cells that are going to start pumping out antibodies, and those antibodies are going to circulate in the bloodstream. Antibodies are these Y-shaped molecules. We can produce a whole huge assortment of them. If you take an immunology class or something like that, they'll talk about the mechanism by which you make them. It's actually pretty cool. And when you have something that has antibodies bound to it, that is a target to macrophages to destroy. There's also sometimes a problem where your body will accidentally produce antibodies against your own proteins. Usually when your immune system is functioning, it can distinguish your own cells from other cells. Sometimes there's a malfunction and your antibodies will attach to something within your own body. When that happens, your immune system attacks yourself. That's called an autoimmune disorder. The cell-mediated response is basically it is going to attach itself to an infected cell and kill it. When you have a second exposure to any given pathogen, you're not only going to be able to mount your response against it faster, you're going to have a much stronger response. So it's going to be like, oh, it'd be like, let's say we had one of the hall monitors. There was a trespasser in the school. The first time they were like, hey, get out of here. The next time they see them, though, they recognize them faster. It doesn't take them as long to realize, hey, you don't belong in here. And instead of saying, hey, get out, they like point a flamethrower at them. Much stronger response and much faster response. But this is different for every pathogen you encounter. So notice up there we have two lines, a blue line and a purple line. The blue line is exposure to antigen A. The purple line is exposure to antigen B. Just because you have a faster response to antigen A doesn't mean you have a faster response to antigen B. That is a whole separate thing. You have to have the right helper T cell activated, which then has to go find the B cells and turn them on. So that's actually the reason why we get vaccines. Vaccines act as that first response. Your immune system has a chance to have a helper T cell react to that, go on and recruit some B cells, have some memory B cells in place, and then if you're actually encountering that disease, your body can fight it off so that you don't get sick in the first place. Now, for vaccines, they used to use like a destroyed version of the pathogen, maybe like an empty virus. Now they can just use like some of the molecules from the coat of it, so they don't even have to infect you with the actual pathogen. Speaking of viruses, we never got to talk about them, so let's take a side trip and talk about viruses. Viruses are not alive. Viruses don't metabolize, they don't take in food, they don't produce waste, and they can't reproduce on their own, so they are not considered living things. They do, however, hijack cell machinery in order to reproduce themselves, but they don't have any of the necessary molecules, enzymes, organelles, anything. They don't have ribosomes, they don't have RNA or DNA polymerase or anything. Yes, they are rather creepy. Fun times. Okay, so viruses, I'm sorry, the formatting on this is not good. I will fix it for next year. So what viruses do is they don't actually physically enter your cell. What they'll do is they will stick their DNA, or in some cases their RNA, into your cell. That DNA is going to have to get past whatever cell defenses you have, like ribonucleases that are going to go and like eat any foreign RNA that's in your cell, and they will become part of the host cell's genome. When they do that, they'll be like really easy for RNA polymerase to bind to. RNA polymerase will attach, start transcribing the genes, which will then go out into the cytoplasm and be translated by ribosomes. Once that's done, then these things will be assembled by the cell's own machinery and used to make new viruses, which a lot of times will lyse or pop the cell on their way out. They can either like bubble out, so you have constant production of viruses, or you can have them just completely destroy the cell and explode outward. Viruses don't move under their own power. When they're not in a cell, they're not doing anything. They're just complex molecules. I could take a virus, I could stick it on the surface of the moon. It's not going to do anything. It's not going to change. On the other hand, if I took a bacterium up there, there are chances it could adapt to be able to maybe survive a little bit. I'm not making grand promises. I don't want you to think we left space bacteria up there. But viruses don't have that capability. However, when they are inside the cell, when their DNA is inside the cell, it can actually mutate fairly quickly, and that's purposeful because the faster a virus can mutate, the more successful it is at spreading things. So viruses are not alive, but they do evolve. They do change because they do have mutations. So when mistakes are made copying viral DNA to stick into a mature virus, that could end up being a virus that's able to infect more cells, that's able to evade the immune system. And so because of that, that virus will be able to evade the immune system, infect another cell, and produce more offspring, for lack of a better word, viruses. So it will be more reproductively successful. All right. Viruses can have a lot of genetic diversity. There are a couple of ways that they can actually change. So, usual places you have any sort of mutation are going to happen with viruses. So, DNA replication, transcription, you might even get a mutation. They don't have anything to check for errors, so they don't check their DNA or RNA for mistakes. And they do have some pretty high rates of mutation. If you have viruses that are kind of related to each other, they have similar enough structures, and if they infect the same cell, they could actually combine forces to become a much worse virus. In fact, that's where like avian flu or bird flu came from. It was a bird virus that mutated to the point where it could infect humans. Same thing with swine flu. So that's why you have to be really careful. That's why, for example, you're not supposed to use like cat poop as fertilizer because they are domesticated. You know, my environmental self has thought about that. I have plenty of cat poo. Let me fertilize the lawn. And then people are like, no, because apparently they are able to transmit viruses that we can catch fairly easily since they've lived in such close contact for thousands of years. And because of that, if you grow your food with their poo, then they could carry viruses that could infect you. Yes, I know it's not fun to think about, and I would wash any vegetables I grow, but still. HIV is the kind of poster child for rapid viral evolution. It is an RNA virus which does not have as many ways to kind of go back and check your DNA. And it evolves very rapidly. It has a very high mutation rate, and because of that, it was very difficult to identify, it was very difficult to treat, and that's why there's no cure for HIV, because you could spend a couple of years curing a strain of HIV, but by the time you do, that may not be the strain that is infecting people. And the problem is, once viral DNA is inside the body, it's very hard to get rid of. Your cells, somewhere in your body, probably carry the DNA somewhere of every single virus you've been infected with. That's a possibility. It's not a certainty. So for example, how many of you guys have had the chickenpox? Y'all got the chickenpox vaccine, didn't you? Young people don't know what you missed. It was terrible. It was terrible. So I had the chickenpox when I was like 2. In fact, I still have scars from it because apparently I wouldn't stop scratching. And because of that, I have chickenpox viral DNA in my cells somewhere. When I get older, if I live to 70 or 80 and my immune system starts getting weaker and it's no longer able to recognize pathogens, that viral DNA may activate and start infecting other cells in my body, in which case I'll get something called shingles, which is just adult chickenpox. Yeah, sucks. All right, so reproductive cycles of viruses. You can use viruses to transmit genetic information. There's a process called transduction, which you see on the right. That is where you use viruses to insert genes into bacteria. Viruses are designed by their evolution and selective pressures to get their DNA into a host cell's DNA, so they're great tools for that. There are also these segments of DNA called transposons that allow chunks of DNA to jump into or out of host cell DNA, and we're able to use some of those for some genetic engineering techniques. This is the scarier part of the virus life cycle. It was scary enough when it was blowing up your cells, but this is when it's a sleeper agent. So the lysogenic cycle is, we're not 100% sure what triggers a virus to start making your cell make little baby viruses. It depends on the particular virus, it depends on the conditions. And so you have some cells that can inject their DNA into some viruses that can inject their DNA into your cells, it becomes part of the host DNA, and then it sits there. In cases like with the chickenpox shingles thing, it can sit there for 30, 40, 50, 60 years. And then at some future time, due to some signal, it'll activate. And then that virus will start replicating and it'll go into what's called the lytic cycle. And that will make you sick. Kind of sucks. All right. Our last major topic, the nervous system. So, neurons are the basic unit of structure and function. Neurons have several parts. The most important parts are the one labeled 1, which is the dendrite. That receives information from other cells. The axon is the output that sends information to another neuron. The cell body is where any signals from all the different dendrites come in, and a decision is made, do we send a signal or do we not send a signal? Notice that this nerve cell has a lot of dendrites, so nerve cells can get a lot of different inputs, but it only has one output. It only has that one axon. So nerve cells take in a lot of information, and then they decide whether to send a signal. There are some structural adaptations that let you speed up the transmission of signals along a neuron, like myelination. It's a fatty sheath that surrounds an axon, and what it does is it makes the signal that's traveling along a neuron jump from one gap between these pieces of myelin to another. So instead of an action potential traveling all along an axon, it can jump from spot to spot to spot and skip all the spaces in between. And that's important because you have some long axons in your body. You have one that goes from the base of your spine all the way to the tip of your toes. So, and that's not very tall on me, but that's pretty tall on somebody who's like 7 feet tall. No, you have multiple, because you're going to have sensory receptors going out to your toes, to the skin of your toes, so you can feel things, and you're going to have plenty of those. You've got a number of them for every square millimeter of skin. Then you're going to have motor neurons that are going to go to the muscles in your toes. So there's plenty of neurons. You don't have just 2 that are long like that. And so, yeah, I don't think need to know nodes of Ranvier or the term saltatory conduction, but you may need to know that this myelination speeds up the transmission of what's called an action potential. An action potential is the way a neuron sends a signal. So it is actually an electrical signal, and then at the very end of the neuron, when it's sending a signal to another neuron, it's going to change to a chemical signal. So the electrical energy is carried just by charged particles, just like electrons carry energy in your electric devices. Ions, which are charged atoms, are going to carry the electric charge inside of your neurons. Sorry, he was trying to eat my ID badge. So this is all due to the concentrations of different ions inside and outside of the cell. So what you'll notice here is that there is a lot of potassium inside the cell and very little potassium outside the cell. If you opened a gateway for potassium to pass, what direction would it move? To the inside or to the outside of the cell? It would move from the inside to the outside. Remember, it diffuses from where there's a high concentration to where there's a low concentration. So it would move from where there's more to where there's less. What about sodium? Where would sodium move? From outside to inside. So if I opened up a gateway in a cell membrane to allow sodium to move, it would rush into the cell. What about chloride? It would move from the outside to the inside. And I cannot ask that question about the last category because those things are too large to move through the cell membrane. They're like really large proteins, and so it would be hard to get them in and out. So a couple of terms. The potential of a neuron is its voltage or its charge. It can be positive or negative. And if you are increasing the potential, making it more positive, that is called depolarization. If you are decreasing the potential or making it more negative, that is called repolarization. And that's important because we're going to talk about how depolarization and repolarization are part of an action potential. So normally neurons are pretty negative. They're negative 70 millivolts. You don't need to remember that number. But what you do need to remember is how molecules move in and out. So what you see here is you see the 3 major proteins that are stuck inside the membrane of a neuron that are important. The sodium-potassium pump is constantly building a concentration gradient. It's funneling sodium to the outside of the cell, and it's bringing potassium inside of the cell. So that way, if a sodium channel opens up, sodium can rush into the cell. Keep in mind, can ions pass through the cell membrane? They need a carrier protein, which is this channel. They're too charged, so you're right. Potassium also can't cross unless it has some sort of carrier protein, like this channel, and when those channels are open, potassium rushes outside of the cell. So what ends up happening is when you have positive ions come in, that's going to make the charge more positive. That's going to raise the potential, it's going to depolarize the cell. At a certain point, that's going to cause what's called an action potential to be generated. And it's this rapid change in charges. So what ends up happening is you pass an action potential along a neuron from the cell body all the way to the other end. This is going to be made possible by what are called voltage-gated channels. They open in response to changes in voltage. So if a cell becomes rapidly depolarized, they'll open up and they'll allow sodium or potassium to pass through. And then sodium-potassium pumps will then restore everything back to its original concentrations, which is important because you want to be able to send multiple signals. So this is the general shape of a graph showing the voltage or the membrane potential of a neuron during an action potential. That dashed line is 0. Anything under that is negative, so you see the resting potential is negative. And everything above that line is positive. So an action potential is a rapid positive change in charge. Dude, buddy.
Oh no.
That is a poopy rabbit. It's okay. Oh, it's still warm. Okay. All right, so let's look. These are the steps of an action potential. What first happens is at resting potential, most of the sodium channels that are voltage gated, most of the potassium channels that are voltage gated, they're all closed. And I like this diagram a lot. It shows you what's happening at each stage of the action potential, and it also shows you that with the graph of the change. So notice in this diagram, you have sodium and potassium channels both closed. When you generate an action potential, something positive charge makes a voltage-gated sodium channel open. Sodium starts coming in. It starts making the cell more positive. This is called the depolarization, and that's where it's at. So you have noticed that the sodium channel on here has opened up a little. Sodium's allowed to come in, but not all of the sodium channels are opened yet. Instead, when you hit what's called the rising section of the action potential or the depolarization, sodium floods in and it causes this massive spike in the charge. After that, the sodium channels start closing. After a certain period of time, they close. Doesn't matter how high the voltage is. Potassium channels, though, are on a bit of a delay. They are triggered by that same depolarization, but instead of opening immediately, they open like a few milliseconds later. And then potassium flows out, which causes the charge to become more negative, and that takes you back to your resting potential. So notice at this point, the sodium channels have actually been plugged up, so nothing can go in or out, but the potassium channels are open, so potassium can leave. There's actually a point at which your charge becomes more negative than resting potential. It's a very short period of time, while the voltage-gated potassium channels are closing up. All right, So those are all the stages of an action potential. There's something called the refractory period. It is very important because it's the period of time during which you cannot trigger another action potential. It's because after they've been opened, sodium channels cannot be opened again for a certain number of milliseconds. Also, that hyperpolarization, that extreme negativity, also prevents an action potential from being set off, which is good because without it, you would actually have action potentials traveling both ways along an axon. Information flow in a neuron is very one way. It comes in through the dendrite, it goes through the cell body, it leaves through the axon. That's the only way things travel. If you have some other direction that information is traveling, you've probably got a disease of some sort. I also like this diagram. It's very similar to what I have in your interactive notebook, where it's showing you the changes in charge as you move along an axon. So on the left side, that's closer to the cell body. On the right side, that's towards the next neuron in the chain. So you have an action potential come in, and it makes that first segment positive. That positive charge spreads, and so sodium channels get opened up in the neighboring area, which is going to make it more positive. Potassium channels in that first area have finally opened up, and they restore the negative charge. Then, finally, the action potential travels farther to the right. The sodium channels behind it are closed. They can't open for a certain period. So only the potassium channels are changing the charge, and they make it negative again. After this point, you could probably have another action potential come down. So you can have a series of action potentials, and keep in mind, this all happens in thousandths of seconds. So you can actually stick an electrode next to a neuron and record its action potentials. It sounds like a bunch of pops. It'll go pop, 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 pop. I mean, they'll be very rapid. An action potential is all or nothing. You either fire or you don't fire. What gives information is the pattern of them. And also what gives information is what happens at a synapse, which is where two neurons communicate. So the end of an axon is called a synaptic terminal. At that point, you change from an electrical signal to a chemical signal. So what happens is there are a different type of channel now. It's a voltage-gated calcium channel. When it opens up due to positive potential, it lets calcium in. Calcium is a very important second messenger, but more importantly, it allows these things called synaptic vesicles to bind to the membrane. These things are bags full of neurotransmitters, and neurotransmitters are the chemicals that are going to send messages to the next neuron. When that neurotransmitter is released, it diffuses across this tiny space called the synaptic cleft, hits a receptor on the next neuron, and that's going to cause some potential changes in that neuron, which can then cause it to fire. There are a lot of different chemicals that can act as neurotransmitters, and the receptors they act on can be different as well. Some of them are just straight up ion channels. As soon as a neurotransmitter binds, they open up, let some sodium come in. Others are actually G protein coupled receptors, so they can set off other signals. They could even set off multiple signals, like one G coupled protein receptor could open an ion channel and turn on a specific enzyme that will then cause a series of changes that make you make different DNA. These are some of the common neurotransmitters that may be mentioned on the test. They can be put into two categories, excitatory or inhibitory, meaning they're either going to try and activate the next neuron in the chain or they're going to try and deactivate it. The excitatory ones, out of them, acetylcholine is important for muscle movement because that's the one that's released at the neuromuscular junction. Epinephrine and norepinephrine, also known as adrenaline and noradrenaline, are related to they also act as hormones. They're related to the fight or flight response that you have. So if you've got an adrenaline rush, your heart beats faster and all of that so that you're prepared to fight or run. Dopamine and serotonin are related to mood. Dopamine is released whenever you do something or see something that causes a pleasurable reaction. Like if you make a bet against somebody and you win and you're like, yes, I'm awesome. That's dopamine release. Serotonin is very, very highly linked to depression. So problems in serotonin release or problems in serotonin transmission are often the targets of antidepressants. GABA is just an inhibitory neurotransmitter. It kind of calms down activity in the brain so that you don't get too much firing. If you don't have that inhibition happening, you can actually get so much firing you get a seizure. So let's talk about the brain. So the brain can produce some neurohormones. So these will travel through the bloodstream and they'll have an effect elsewhere in the body. The brain has three major sections you can divide it into. The cerebrum. Okay, I misplaced those. It's supposed to be the cerebrum is the forebrain. That's got the cerebral cortex that takes in sensory info. The brainstem is the midbrain. It controls stuff that's not voluntary. So that can control your breathing, and it also sends information higher up. The cerebellum is the hindbrain. It's literally named little brain, and it's involved in coordinating movement. You have a structure deeper in your brain called the hippocampus. It's involved in forming long-term memories. So when you damage that, you'll get people like, have you ever seen the movie Memento? It was really big when I was in high school. This guy loses his ability to form long-term memories, so he tattoos everything on himself so he doesn't forget. Then you have another region that also involves the hippocampus. It's called the limbic system. It's like called emotional cortex because this is where you process a lot of emotions. It's attached to the hippocampus so you can add emotional context to your memories. And it also contains something called the amygdala, which is what causes fear responses. So for example, if you could start stimulating someone's amygdala, they would get scared for no reason. The different parts of the cerebral cortex have different sensory data that they get. You can see on this diagram the visual cortex, which takes in visual information from your eyeballs, auditory cortex, which takes in information from your ears. One area that's not labeled on here at the very front of the brain is the prefrontal cortex. That's involved in decision making. And if you get a chance, Google Phineas Gage, Phineas Gage. He got he got a steel pipe, accidentally exploded and shoved up through his chin and came out the top of his head. And he was actually able to live past that. The thing is, it damaged his prefrontal cortex and his personality completely changed. And that was some of our first evidence that the prefrontal cortex controls elements of personality. You have two hemispheres, a right hemisphere and a left hemisphere. There's some specialization, and also they process body information for the opposite side. I've never found out why, because it seems weird to me, but there are some people who are occasionally born where it doesn't cross over. So the right side of their brain controls the right side of their body, and they usually have some pretty bad defects. So they may not even be able to function very well. So right side of your body sends information to the left side of your brain and vice versa. There's some specialization. So for example, all of the information you're getting from your ears right now, since it's in the form of speech, is being processed by your left hemisphere. If people have damage to their left hemisphere, they may lose the ability to understand or produce speech. But if they damage their right hemisphere, they may not have that same problem. The two hemispheres have to connect. They're connected by this thick tract of axons that's passing back and forth between the two hemispheres, and it's called the corpus callosum. And this is just for fun. It's called a homunculus. It's a representation based on area and how much of the brain area is devoted to either receiving information from various senses or sending information to various muscles. So the size of the body part reflects how much representation it has. So notice on the right side, the sensory homunculus has a huge spot for the head because you get a lot of sensory information from your face. Your nose, your lips, your eyes. If something's touching your face, you want to know. On the other hand, if something is touching you on some place like your back, you don't get a lot of sensory data from that area and your brain doesn't pay as much attention to it, so it's just like, yeah, whatever. Motor control, notice that on the left-hand side you have a huge face, a huge region for face, lips, jaw. That's because we make a lot of facial expressions, and those are controlled by a lot of muscles, and we have very fine control over that. And that's it.